I'm going to make a start and introduce Joëlle and the next exciting seminar in our exciting seminar series. So welcome back, welcome to 2024, and we are sure this is going to be a very exciting year. This is a second FCS themed seminar in our exciting seminar series, and it's really becoming very obvious that FCS is a cutting edge technique giving really unmatched insight and resolution into molecular characterization. It's a method that's not just for academic labs, but there's massive applications across pharma and biotech as well, and showing it's really translatable in different ways. And some of Joelle's work will really discuss some of that around drug stimuli and mapping effects of this in using FCS. So I'm really looking forward to Joelle's talk for those reasons. And as ever, of course, you know, if there's anything that comes out of this and you're like, oh, I'd like to do that experiment, have a chat with one of us or our application specialists, and of course, ask questions. And so the way to ask questions throughout the talk, if you type them into the chat, that will work best. And then we'll pick those questions up at the end of the talk. So over to Joëlle. Joëlle graduated from University of Nottingham in genetics, in fact, wasn't it? And then went on to complete a PhD in related areas into the Institute of Genetics, and really her postdoctoral work, you know, is sort of stem cells and cerebellum sort of areas. It wasn't until moving to a role at Nottingham in 2007 where she kind of changed fields a little bit, with more of a focus on molecular pharmacology. And it's in that work, and then went on in 2009 as a visiting academic in Würzburg, using flash-based FRET technique for GPCRs. And I guess that was where a lot of the starting sort of things for this work really came into being. In 2015, she was awarded an advanced diploma by the British Pharmacology Society, and is now research fellow in advanced microscopy as part of the Compare network here in the UK, which is a joint institute, I guess, between Nottingham and Birmingham. Is that right, Joëlle? And she's doing a lot of work developing fluorescence correlation spectroscopy alongside other methodologies for the study of membrane receptors with other imaging technologies. As I say, questions in the chat as we're going. We'll do them all in a one-er at the end. And Joëlle, really looking forward to your talk. The floor is yours.
Fab, thank you very much. Now I'm going to see if I can actually share my screen.
I think I stop and then you start, and the magic happens.
And then if I can actually find my talk, which I have. And let's make that slideshow. And I think I need to swap. Brilliant. Can you now see my title slide?
Perfect.
Brilliant. So I'm going to move straight on. Actually, let's change my mouse into a laser pointer. So thank you so much for the introduction, Tim. I suppose a little bit more about me. Why not introduce myself again? So yes, based in Nottingham, where I work does actually look as beautiful as the bottom right corner, but this is the picture that we tend to kind of put out there as a come to Nottingham and our beautiful campus. But I am part of the cell signaling research group, and we're all actually part of a large consortium, as Tim mentioned, Compare, so the Centre of Membrane Proteins and the Receptors, and we're all passionate about trying to understand how membrane proteins are organised on the cell membrane, how they function, what they do after their stimulation, and typically applying a lot of microscopy-based techniques to understand this. And so the nice little graphic in the top right shows a lot of the different things that we are applying to try and understand these membrane proteins, trying to understand the pharmacology of these, and therefore the bigger aim to design and develop better drugs in the future. Now I'm trying to move on my slide. So this is where we tested moving the slides and it worked beforehand. There we go. So I've structured my talk in 3 main parts. I want to introduce to you why we would use FCS to study these membrane proteins, why it's such a useful technique. Then I want to move on a little bit more to my kind of passion, is trying to move towards physiological relevance. So how can we actually study these receptors in their native environment, and how can we move away from them all the model cell model cell types that we're using, which tend to be very much away from what we can see in humans. And then finally, I want to bring in using fluorescent ligands alongside these. And so basically a whole kind of swarm of different techniques that have come together with FCS to try and push our science further. So FCS for studying membrane proteins. What are membrane proteins? I know the audience might be quite diverse. So membrane proteins, they're basically the proteins that sit on the cell membrane. We can stimulate these proteins with a ligand or a drug, so anything you can get over the counter, and these will then receive the drug and then signal within the cell, dictating what that drug is supposed to be doing to the cell. So these membrane proteins are kind of like the middlemen of what's going on with this action. So that's really important to understand how the membrane proteins function, because if we can understand how they function, we can drug them better. A large family of these membrane proteins are called the G protein coupled receptors. So these are, I suppose, I think it's about now 35% of all drugs are targeting GPCR. So they're incredibly important. They're targeting, and I'm going to go on and talk about them in my talk. So drugs, for example, that you're using to treat asthma, so cardiovascular diseases, lots of neurological type diseases as well. So whilst we've got a great amount of these drugs, they're not perfect. They can have lots of different types of side effects. The selectivity might not be great. You might not be able to take them if you're suffering from another type of indication. You might be taking different types of drugs, and these aren't going to work together very well. So there's always need new investigation into how these drugs work, and therefore we really need to understand what are the receptors that they're targeting. So these receptors, when they're stimulated, they let out a signal into the cell, but they're also controlled after this. And how they're controlled can vary quite a lot between the different types of receptors. So you can have these receptors move together. They can be internalized. So I mean, they're brought into the cell, ready to be recycled. Some might be turned off by being moved into the cell or by changing the conformation. So the more that we can understand, the better we can then design some of these drugs. How do we label them? So if we want to investigate them via imaging, which is one of my main aspects, we need to label them with something fluorescent, something that we can detect. So here is my example of a GPCR again. It's sitting in the lipid bilayer. This is the outside of the cell. This is the inside of the cell. Quite commonly, we can label them with a fusion protein. So these are typically well known, like GFP. We can stick it on the inside. We can stick it on the outside. We need some genetic modification to be going on. These are great because then we know that they're always going to be expressed when you're labeling something with a GFP protein. Another option is by genetically engineering a protein tag that instead of, let me see if I can move on. Right, I think I'm going to come off my laser pointer because that is making it impossible for me to move on. So sorry about this, give me 1 second. There we go. Let's just go with the mouse button, if you can see my mouse. There we go. So we can engineer a protein tag. This protein tag is now sticking outside of the cell, and then we can add a dye to this protein tag. The benefit of this type of application is that you can choose the color, and there are a number of different genetically engineered protein tags, so you can multiplex these as well. These are great for looking and studying what the receptors are doing, and you can actually choose how much you label them by titrating in the dye, so you're not labeling everything. The negative with using a fusion protein is that it's always on. You may have too much going on. This may be negative to what you want to be doing in terms of your microscopy. And the third option is using a fluorescent ligand. So when I mean a fluorescent ligand, I mean it's typically a drug that has had a fluorescent tag attached to it, which will selectively label the receptor that you're interested in. And there we go, it can come along and stick onto the receptor of choice. So once we've labelled our receptors, there are multiple different ways that we can study them using microscopy, and we just study their dynamics and study how they move on the cell membrane. These can include FRAP, which is when you can bleach an area and you can see how quickly that bleaching recovers, so you can get out the speed of movement of these receptors. We can also look at single molecule tracking, where we can follow individual receptors, for example, like turf, and we can understand how they're moving on the membrane as well. And there's another technique called RIX. Again, this is very similar to SBS, which are going to come on along in a minute. And this can look at on an ensemble field of view how fast the fluorescent species are moving. So these are all great, and I think, to be honest, with these types of techniques, it's all complementary to build a bigger picture of how to study the dynamics of your receptor. However, they do have slight limitations. So for single molecule tracking, you're relying on all your receptors being in one plane. You're relying on them not getting bleached too fast, because otherwise they're suddenly going to disappear. For FRAP, we can only actually define one average species that's moving within that field of view. You're getting quite a large area that you're bleaching, so it's not necessarily on the nanoscale. We're looking at a more of a macro scale image of what's going on. So to combat this, or to complement this rather, use fluorescence correlation spectroscopy. And I know maybe the audience may or may not be totally au fait with FCS, so I'm going to give an overview but not go into too much detail. So with FCS, we can have this observation volume that is formed by a very standard confocal microscope, and this observation volume can be placed on a position within the cell. For example, the cell membrane inside the cell, it can be placed in solution. And what happens when your fluorescently labeled receptor passes through the volume is that it gets excited by the laser, it lets out light, and that light can be detected by your detector, by your camera. And what you tend to do from this point, this observation volume that stays still in time while all the fluorescent species move, you get out this fluctuation trace. So it's a fluctuation of intensity over time. And by using some clever maths, which I'm not going to go into because, to be frank, I don't quite understand the maths. Every now and then I get it, but I want to understand more of the applications of FCS. You can autocorrelate these signals to pull out this great curve, which can give us the concentration. So the y-intercept gives you the concentration of the fluorescent species at any given time, and then the half dwell point of this curve will give you the time that it takes for that fluorescent species to pass through the confocal volume. So you can see how this can be incredibly useful to understand a concentration of your biological species and also how fast it's moving. This is quite a nice graphic from I stole from the PicoQuant site, because actually if you look at this y-intercept, this intercept is inversely proportional to the concentration of your biomolecules. So as you increase the biomolecules, this curve goes down. So FCS is actually a lot stronger of a statistical technique with the lower concentration that you get. So as your curve goes down, you've got a higher concentration. And as your molecules get bigger or are moving in a more complex fashion, this curve moves along to the right and you get slower diffusion. So we can get quite a lot out of this curve. However, they don't always look that good. This is a curve taken from an autocorrelation curve taken from a cell-based reading, and it gets a lot more complicated. So you do need to understand your biological system to actually understand what you can get out of it. So the beauty also is that you're getting intensity-based readings from these fluctuation traces. And if we apply a slightly different analysis to this trace, so if we cut up our trace into little bins of time and plot this in a histogram called a photon counting histogram, we can actually get an understanding of what the average molecular brightness of your fluorescent species is. And so if you have a standard of a 1 brightness or standard of 2 brightnesses, we can then take your experimental sample and understand a little bit more about the stoichiometry, how many fluorophores are in the average particle that's passing through your confocal volume. And so this is an actual trace. I think this is probably a fluorescent dye in solution, and this is a typical curve that you can pull out, and you can pull out the brightness. So why would FCS then be useful when we want to understand protein dynamics? So if you're thinking about diffusion speeds in a physiological setting, the cell membrane is really complex. So the more complex it gets, you can imagine the more difficult it is for the membranes to move smoothly, for the receptors to move smoothly in the membrane. So by changing diffusion speed, we can understand a lot more about the dynamics of our receptors. The concentration of biomolecules is really important. So as these receptors are regulated, as they're internalized, as they are upregulated, that concentration is going to change on the nanoscale, and we can directly pull that out. And indeed, like I said about the brightness, you can pull out the stoichiometry of these complexes as well. So are these receptors clustering over time in response to treatment with a drug? And especially what I'm keen on understanding is the nanoscale dynamics of these receptors, and the fact that we can resolve multiple diffusing species. So other than, for example, FRAP and with RIX, which is looking at more of an ensemble average, you can only pull out a single diffusing species. So if you've got a couple of diffusing species within your cell membrane, you're just going to get an average out. Whereas FCS has the power to separate out those diffusing species. There is a limit, obviously, but then you could understand maybe what populations and how those populations are changing over time. So I just wanted to bring in a few examples of this actually in practice. So these examples are work done from people who've come PhDs and postdocs over time within our group. And here we've got a typical GPCR, an adenosine GPCR. So adenosine, very important in the treatment of cardiovascular diseases and the immune response, and also with neurological diseases. And here we've got a fusion protein stuck on the intracellular side. So this receptor can be expressed in a model cell line. Here's a CHO cell line. These is just focal imaging. And we can pull out really nice traces from the membrane reads. By applying the autocorrelation analysis, you can get out these curves, and then you can start to understand what how fast the receptor is moving. This first curve can be modeled by 2 diffusing species. And now this is where you have to get a bit wise on how you set up your system. So we know that GFP has some photophysics to it. So we know that this species here, this moving species, is way too fast to be a membrane receptor. What we do know it is most likely going to be the photophysics of GFP. Whereas this much slower point here, so this is probably in the tens to the hundreds of milliseconds, is going to be our GFP labeled receptor, and we can pull out diffusion coefficient of up here, it's 0.1 micrometers per second. So we can use FCS with our model system to understand what our receptor is doing. And that's all well and fine, but actually what we care about is then sticking on some drugs and seeing what effect this has. So I'll just move on to my second example. So this is work done by a PhD student, Arabelle, who came and worked with us, and she was interested in the mu-opioid receptor. So another GPCR, and this one, opioids are very much involved with the treatment of pain, but obviously as well, there's a lot of misuse of opioids. And the problem with opioids is, whilst they treat pain, they can also dampen your respiratory system. So there's a lot of instances where we want to design better drugs that are going to treat pain, but not going to have the side effects. So to design those better drugs, we need to understand how the current drugs are working. So for example, the mu-opioid receptor can be treated with morphine, and it can also be treated with another drug known as Damgo. So these 2 drugs differ slightly with how they work. They both will stimulate the mu-opioid, but they have different methods of functioning. So this confocal image here at the top, this is I think these are hex cells. I think these are hex cells which have got expressing this muopio with a snap tag, which is on its the outside of the cell, and it's been labeled with a fluorophore and things, selective fluorophorate. And you can see where the receptor is localized within the cell. When it's treated with these two different drugs, the damgo drug will cause the receptor to internalize, to go inside the cell. So too much stimulation of the drug turns off the receptor, moves it inside the cell, whereas morphine doesn't have this effect. So they are functioning in a different way. So what Arabelle did was that she did some FCS on this receptor, looking at two different drugs to see what she could see actually going on at the single receptor level. We can see what's going on the mass of the cell, but we can't see what's going on the single receptor. So these are the traces that she pulled out, and immediately you can see that this trace of this damgo, the internalizing one, has a lot lower count rate, which immediately says to me there's a lot less of that receptor on the membrane. These are the autocorrelation curves she pulls out, and again you can see the one in red, the damgo, is y-intercept's a lot higher, so this is a lot lower concentration. So what you could see, what you could pull out, as I said with FCS, you can pull out how fast these receptors are moving. So she looked at the normal condition, normal receptor under vehicle. She looked at the effect under the two different drug stimuluses. And she could see that the diffusion coefficient of damgo was less than from morphine. So something's going on with treatment of this drug to the receptor that isn't happening with the morphine. So it's slowing it down. She looked at the particle number, and also, as we could see from the traces, this particle number is decreasing. And then finally, she looked at the brightness. And what I want you to look at here is the fact that 53% of these really bright particles, these really bright single particles are being detected. So these are super bright. So this is indication of some clustering. So what SCS was able to do is, we can understand that these two drugs have different ways of functioning just by looking at their confocal imaging, and also maybe looking at some downstream signaling. But what is actually going on at the membrane, what's actually happening in terms of organization? And what Arabelle concluded was that these receptors, the receptor and the stimulation of damgo was clustering. So these clusters, it was being slowed down, whether or not this is just getting into a more complex situation on the membrane, or whether it actually was slowing down because there's more of it, and the particle number was decreasing. So FCS is really powerful for bringing out those elements that you might miss otherwise. So that's how FCS can help protein dynamics. Now, what do I care about? What do I want to push it towards? So I really want to move towards physiological relevance, because those two examples I've shown you already are from systems that are overexpressed, and they're in model cell lines, they're in CHOS and they're in HEX. So how can we move towards physiological relevance? Well, we could use stem cells. We can use cells that have come from, especially human stem cells, which we can then push to produce different cells within the body that are going to more confidently reflect what's actually going on. And so when we study what effect the drugs have, we can get a better idea of what's actually going on and what will be more useful for us to develop in the future. So this was a project that I was on, and in collaboration with a great scientist, Sasha, who was able to create different haplotypes of this beta-2 receptor. So the beta-2 receptor was really well known in terms of drug treatments. So cardiovascular, respiratory, many people suffering from asthma, your bronchodilators will often have a beta-2 agonist. We can also, if you've got a bit of a fast heartbeat or a bit of blood pressure, you're probably taking a beta blocker. So really useful to understand how these drugs work and to improve them. So the receptor within the human population has a number of different polymorphisms. A couple of these arise from these single nucleotide polymorphisms on the N-terminus. So two of these, potentially 27 and 16. So if there's two different sites, you can think that if we have all the different combinations, you can get 4 different haplotypes in the human population. And these very much depend on your race, as in which one, high race weighting, if that makes sense. But and they also thought that the haplotypes could have affected how well you receive different types of cardiovascular drugs. So the question for this project was, what does the effect of the haplotype have on agonist-induced receptor internalization, and how can we go about studying these? And what Sasha was able to do was create me some cell lines which had beta-2 with a snap tag on it on its external side in stem cells, and he was able to differentiate these into 2 different cell lines, fibroblasts and cardiomyocytes. And this is important because cardiomyocytes, as you may know, are terminally differentiated cells, so they're not going to replicate, they're not going to divide in culture. So it's really hard for us to test how these drugs work and test how receptors are functioning in the cell, in the lab, like you can do with a model cell line. So what we need to use, what's really useful to use is cells, cardiomyocytes that have come from stem cells, because we can create these without having to get cells from biopsies and so on and so forth. So what we did was to look at these, this receptor first off on the confocal microscopy. So here we've got some stem cells. You can see the beta-2 with a snap tag. On the left-hand side, vehicle conditions, normal conditions, and then we stimulate with a drug and we can cause this massive internalization of the receptor. So the receptor is being moved inside the cell in response to a high concentration of drug. And because this is so beautiful on the confocal, we can use just basic imaging to count how many clusters there are and pull out these graphs on the right. I'm not going to go through this too much, but these are the four different haplotypes that are possible, and the different colors of different drugs. And what you can kind of see is this top left one, this RQ haplotype, you're getting higher curves and you have the other haplotypes. So there seems to be some haplotype-based effect on how much receptor gets internalized. Oh, I think this is great. This is going to be really easy. We're going to push our stem cells into fibroblasts, cardiomyocytes, and replicate this, and this is going to be really quick to do. However, as soon as we moved into fibroblasts, the expression levels seemed to drop. We had less, the imaging was harder to do, and we couldn't see any difference after we added our agonists. I thought, fine, let's have a look at the cardiomyocytes, because they're what we really care about. And then when we moved on to cardiomyocytes, we couldn't see them at all in the confocal microscopy. So they've still got the receptor with a tag on it in there, but we can't see them. And therefore, if you can't see it, you're not going to be able to do any confocal microscopy on this. We're not going to be able to measure the internalization. So lucky for me, this is a great FCS project. And so I thought, okay, fine, I'm going to do some FCS on this receptor in primary cells and see what I can pull out. And then now you start to realize why people are using model cell lines, because these primary cell lines are really hard to FCS. They're non-uniform, they're like fried eggs. They're really hard to find the membrane. We can't see hardly anything. So what I had to do is develop a method to find the membrane, and basically it's taking multiple FCS reads at different points. So this is a Z scan. So if you kind of turn your head to the side, this is the top of the cell, this is the bottom of the cell, and as I go down through the cells, I'm taking different trace reads. So this is a laborious microscopy experiment. We can pull out autocorrelation curves, but then we can find the nicest autocorrelation curve and get out our FCS read, which is going to give us information about what's going on with the receptor. So FCS, great, powerful technique, but it is slow with cell-based measurements. So with a lot of work and love, we're able to pull out traces for each of the different cell types, the stem cell, the fibroblast, and the cardiomyocyte. And then we're able to actually pull out some information to understand what's going on, what's the difference. And as we can see by confocal microscopy, the receptors are there, but the amount of them is severely decreasing as we're differentiating them into the different cell types. So the cardiomyocytes here, the numbers are on the right-hand legend. So 20 to 30 particles per micrometer squared, whereas the fibroblasts, you're about close to 100, and the stem cells are close to 200. But what was really interesting to me is that the diffusion that I measured didn't change. So it was really consistent, the diffusion of this receptor between the different cell types. So we can do FCS with primary cells. Can we do pharmacology? We're using FCS on primary cells. So coming back to the stem cells, which you could do the confocal microscopy to look at the internalization, and we had an increased internalization with this one haplotype. So I then looked at the particle number of this same haplotype in the stem cells, and you could actually measure a drop after you treat it with the drug. So yes, we can do pharmacology with FCS. And interestingly, with the fibroblasts, there wasn't a change in particle number. So it wasn't that I just couldn't detect a change with the confocal microscopy. Actually, there wasn't a change. So that was really interesting as well, that these receptors behave differently depending on the different cell types. So agonist-driven internalization of these receptors in response to drug is really cell type specific. And again, if we looked at the diffusion speed, that doesn't seem to change. So unlike Arispel's, where she saw a change in diffusion speed, this isn't changing with the addition of the drug. So it's hard to kind of make a conclusion about what's happening following on from drug stimulation on the receptors, but we can tick off it's not massively changing the diffusion. It's about building up a big picture, isn't it? So primary cells are one way to go to understanding what's going on within the physiological setting. However, we'd still transduce these cells. We still put the receptor in with this tag. Another option is to then moving maybe towards the genetic engineering that we have available to us now, so CRISPR. And this is work I did with Carl, who was able to CRISPR in a SNAP tag onto the external face of the beta-2 adrenoceptor. I mean, we're going back to hex cells now, because you only can do so much at once. You've got to take one step at a time to make your life not too difficult. And you can see, like on focal microscopy, once you CRISPR in the SNAP tag, this receptor is now expressed under endogenous promotion. You can't see anything by confocal microscopy, and they've got the overexpressed cells there on the right-hand side. Beautiful cells you can see on the overexpressed. You can only see a blur. If you kind of squint your eyes, you may be able to see the different cells. But can we FCS them? And we can. And what you can see straight away is the count rate. So the count rate is how many photons the detector is receiving over time. And this is so every time a fluorescent species passes through confocal volume, you're going to get that readout, the excitation readout by the detector. And this is connected to the concentration. So this is much less in our engineered cells. But we can still pull out beautiful correlation curves. And these really are beautiful compared to some that I have seen. You can fit them really nicely and reliably pull out both the concentration and be a lot more confident about the diffusion speed. So here we go, here's the concentration. They are about 10-fold less, the CRISPR ones to the overexpressed ones. And again, the diffusion speeds aren't significantly different. But what I found quite interesting was I also looked at molecular brightness, and these overexpressed cells, on average, the average population was significantly brighter. So something is going on within the cells where we have more receptor being expressed. So there is a thought, especially with these GPCRs, these membrane proteins, that the more there is of the receptor on the surface, the more prone they are to come together and dimerize or form higher order oligomers. And so maybe this is what we're seeing here. So maybe because we're overexpressing our receptors so much, we're getting forced clustering and we're getting brighter species. And so this is almost like a word of warning that there's something going on here. So we've got to be careful with what we take out of our experiments using overexpressed or model cell lines. So both those techniques, though, are still genetic modification. What can we do to look at our receptors using FCS without affecting them at all? So one option is using a fluorescent ligand. And when I'm talking about fluorescent ligand, I'm meaning most often I'm using a drug that has actually got a fluorophore stuck on the end of it. So you've got this pharmacophore end, which will selectively target your membrane protein. And this is for working with GPCRs, which are really highly targeted in the clinical practice. Quite often we're using structures which are very similar to clinically available drugs. We then need a little linker. So this is a variety of different linkers which will give enough space between your warhead, as it were, and your fluorophore. And at the end, obviously, you're choosing a fluorophore, something that's going to be suitable for the technique that you want to use. And quite often we need to think about what type of fluorophore these are, whether they're lipophilic or they're hydrophilic, and how they work. So when we have a fluorescent ligand with FCS, the first thing we need to do, and this is look how this fluorescent ligand moves in solution. And so this is a really useful application of solution-based SCS, is to understand and characterize these fluorescent ligands. The traces are a lot more, you can see this is a really small dye moving through solution. They're a lot more compact, they're a lot more normalized about an average. Those cell membrane ones. And the autocorrelation is a lot faster. This has got probably a 200 to 300 micrometers per second diffusion coefficient. And the nice thing about the fact this has such a different diffusion movement compared to our membrane receptors, which are this is moving about 68 microseconds. Our receptors are in the tens of milliseconds, is that we can differentiate a ligand on its own and a ligand bound to a receptor by SCS curves alone, and we can quantify these. And this was me just reminding myself that diffusion, we tend to estimate the diffusion speed of something based on the Stokes-Einstein equation. So it is really in proportion to its radius, so the size of them. So coming back to my example before, where we used just like the fusion protein to characterize membrane protein, we can now use a fluorescent ligand to characterize membrane protein. And here's an example with an adenosine receptor, which is untagged, and we've got a selective ligand for the adenosine receptor. We first of all need to characterize this ligand in solution so we know how fast it moves, and we know that it's not got a multiple diffusion characteristic, or else it's going to get really confusing when we're trying to analyze our data. And then we can stick it together with our receptor, which is being expressed on the cells, but remembering that we're going to have both bound ligand and also free ligand. And so here's the adenosine receptor, and there you can see it, the confocal image expressed in the membrane. And here it's a lot easier to find those membranes. They're really highly expressed. And you've got, if this is the Z, kind of like a Z position through the cell, if you scanned it at a single point, and this is the lower membrane, upper membrane, and we can place our confocal volume on that upper membrane, pull out a beautiful trace, and here we get a lot more complicated curve. So now in our SCS, in our observation volume over time, in our trace, we are going to have free ligand. But because we know how fast it moves, we can separate this off and quantify it. We can also quantify the bound ligand. And this is this later curve here at the end. Luckily, there's a lot of software out there that does this fitting for you. So whilst this might look a little bit alien to most people, you get to know what these curves look like, and there's also clever software to fit and put your potential models on. So here the bound receptor is about 60% of your curve, and there's also this bit in the middle. So this bit in the middle represents something that's moving about in the 5 millisecond range, and that is way too fast to be a receptor. It's also way too slow to be something that's photophysics or your fluorescent ligand. So one potential is this could be receptor that is being bound by ligand as it passes through the volume, or it's losing its fluorescent ligand as it passes through the volume. So you get an artificially fast accounting for that receptor. So you can see how using fluorescent ligands, we can really pull apart how much free, how much bound, and potentially how much dynamic ligand there is, but that can get complicated. And so for my research moving forward, some things that we've been working on is trying to get rid of this free ligand in solution. So there's the problem of this ligand in solution that might be coming on and off your receptor and complicating matters. And the other big issue is that whilst your fluorescent ligand is on your receptor, this binding site is occupied. So if we care about the pharmacology of a receptor, we've already changed how its formation. We've already changed it because we've got the pharmacophore still stuck in there. So can we actually remove that pharmacophore? And some work, so this is work hopefully going to be coming out soon with amazing chemist Eleonora, who designed this fluorescent ligand. So here's your fluorophore, your linker, and your warhead, which was able to selectively bind this GPCR, adenosine GPCR. But then this amino acid on this tail of the receptor is able to undergo nucleophilic attack with the compound and moves this fluorescent ligand onto the receptor. And if you can do extensive washing, you get rid of that warhead. So now our binding site is free. We can show this nicely with imaging. So here is a receptor that's got a snap tag on it, so we know where it is. You can bind the ligand, it binds nicely to where the snap tag is. If we pre-treat with an antagonist, we stop it binding. So we know it's selective for what we want, our receptor. And then if we allow it to bind again, but then try and chase it off, as you could normally do with a competitive ligand, we can't get rid of it. So it's stuck there. But importantly, can we FCS it? And we can FCS it. So this is the fluorescent ligand in solution. And it's giving us a really nice curve, so we can actually pull out how fast it moves. So then when we can move on to having our receptor expressed in cells, we can pull out a great trace and a beautiful autocorrelation curves, which don't think I have time to go through with now, but have a single component decay curve. This is the photophysics of fluorophore, which is Psi5. And so we're getting no excess fluorescent ligand because we've been able to wash it all out. And we're also getting no additional diffusion speeds, which may be typical of the ligand coming on and off. So this is now allowing us to maybe study the receptor in its true native form without a warhead in place. And this is the pre-treatment with antagonists. It's so clean. There's no trace, there's no binding of a fluorescent ligand when the receptor is not free. And here's some data. So this is a really clean way to get out some data for FCS. I mean, the only negative, I suppose, maybe FCS is this is the kind of data that you're showing people. You're not showing people beautiful pictures unless you can bring in the imaging as well. You're showing them dots and a curve. But really, this does mean something to us. So I just wanted to, I think I've got like maybe 5 minutes left. And so I want to move on. So I really care about receptors and how they're moving on the cell membrane, but that really restricts us to cell-based reads. And as I may have introduced you, it's really difficult to find the membrane of the cell. I haven't talked through all the bleaching issues and the issues with drift of the cell, and the fact it's a really complex situation. What if we could move this to a solution-based answer? And the benefits of solution-based SCS is much faster. You can get reads in tens of, well, you can get reads in seconds rather than maybe 30-second read from a cell. You can also then, if you can have something in solution, we can instantly miniaturize it, which allows us to do automation, high throughput pharmacology, and probably a lot simpler modeling compared to the modeling of a receptor moving on a cell. So how do we get our receptors in solution? So these membrane proteins, you saw they were quite complicated. Without the membrane, they fall apart. And when they fall apart, they're not in the right conformation. They can't be triggered the same way by our drug. They're not going to signal. They're not going to be a good representation of what a membrane protein is. So there are a number of different things we can do. And the thing that we've been trying to push is using these synthetic polymers, these SMALPs, which are able to, you mix them with some cell membrane, and they basically stamp out, pull out little membrane discs, and quite often they'll have your receptor that you've labeled in them as well. And you can purify these, you can play with them, and if you've got your receptor presently labeled, they're the only thing you're going to see in your FCS measurement as it was. We had a PhD student, Rachel, from the University of Birmingham, who came to us, and she was really interested in the adenosine receptor, and she managed to put this in a SMALP particle. So she managed to purify the receptor. And so working with us and a fluorescent adenosine antagonist that we had, she wanted to see whether she could look at the binding of this fluorescent ligand to the receptor in solution within the particle. So first of all, again, you've got to make sure that your fluorescent ligand is doing what it's supposed to be doing in solution on its own, and you know its speed of movement. But then when you add this, the receptor particle in, you can see that this FCS curve moves to the right. So this receptor within its SMALP particle is not labeled. So if we just looked at it on its own in solution, you're not going to see it. You can only see fluorescent things with FCS. It has to be fluorescent. It has to be moving. If it's not labeled, you can't see it. So by moving that autocorrelation curve, we've shifted it to the right. We've definitely bound something bigger. And as we know how fast our ligand moves in solution on its own, we can separate this off in part of the curve. We can fix this. And then what's left would be our receptor and its SMALP. And this is when we back calculated our Stokes science line equation. This is a very representative speed for something of the size that we knew what this size of the SMALP was going to be, so it made sense. And again, the nice thing with FCS is you can quantify free and bound ligand, and we've got a percentage of both. So as soon as you start thinking, well, I can calculate what the binding is, then we can maybe start to do some pharmacology. And so this is what Rachel has done. And so she labeled her SMALP with a concentration of this fluorescent ligand. And by trying to compete off this fluorescent ligand, so as this ligand comes on and off, if you've got a higher concentration of a separate ligand, that's more likely to bind in preference. And by changing the concentration of this unlabeled ligand, you're going to compete off this fluorescent ligand, and you can start to pull out these curves. And these are very typical of the type of binding curves that I like to pull out in industry when you're trying to determine the binding affinity of your unknown. So as a kind of like a screening tool. So you can see how this could potentially be very powerful if we could get our receptors out of their complex environment. So we're not caring about what else is going on in that cell. We don't care that it's a HEC or a CHO. We just have that receptor on its own. We can understand what that receptor is doing in solution. So this is kind of where we want to push with our fun chemistry and our new ligands and our techniques, pushing that forward. So that is it. So hopefully I've put across to you how powerful SCS can be in terms of interrogating membrane protein dynamics, and a few new tools that we've been developing within the group, and therefore the potential of SCS to push pharmacological science, hopefully. So it leaves me just to thank all those who contributed to this work, because it's very much not just me. I may have done quite a lot of FCS in this, but a lot of the other work, the engineering, the chemistry, and also working together with the best molecular pharmacologists, so Steve Britton, Steve Hill, and lots of great colleagues as well. So this work is very much a team science kind of output of understanding what GPCRs are doing, and of course the funding, and also the larger consortium that is COMPARE.
Fantastic, Joëlle. Thank you ever so much for that. There are a couple of questions that have come through the chat, but if people want to kind of raise a hand now and ask things as well, then if you use that feature within Zoom, I'm pretty sure there is a raise your hand option, right? So Ashley will keep an eye on that.
Will I try and stop sharing?
Yes, I guess, or you may want to flick to slides if they arise. So I would keep it on for now, Joëlle, and see what happens. There's one question that came in from Rizal Heradi, who I don't think made it today, but had a pre-question in, and half of his question you've answered, but the other half I will just ask. He says, can FCS measurements of carefully designed experiments talk about the fluidity of membranes?
Definitely, and there's some great work out there as well, which have actually used FCS on cholesterol, which has been fluorescently tagged, and you can definitely pull out how fast that cholesterol is moving. So really, you're sticking in a dye or something that's actually going to stick in a membrane and pull out that stuff. I mean, you've always got to be worried that you're sticking something in, so you might be changing what's going on. Something we're keen to do as well would be to bring in STED FCS, so we can make our confocal volume even smaller, and by changing the size of that confocal volume, you can pull out information about the types of diffusion that's going on, whether the diffusion within your membrane is free, whether it's constricted. But yes, basically you can understand stuff about the fluidity. I think with all of these things, and it's kind of a big push point for me, I think you've got to really use complementary techniques around this as well. FCS will give you one answer, but without a number of different other answers to put together, you don't have the whole picture, and you might be very much biased to what you think's going on.
Brilliant. There were a couple of from me, and then one from Ben Ambrose. So from me, I was interested in how general a couple of these things are. So one, on the downside, how general is it that, you know, when you overexpress these GPCRs, that they're going to kind of multimerize? Is that do you think that's a known problem for these types of proteins?
I think it really depends on the GPCR. There was a great, I think within that, where I actually put it down, let me find the slide, and it will give the paper, which was I doing? Oh, it's my CRISPR slide, wasn't it? That was it.
Yeah.
If I can find it. There we go. So this paper by Isbellit, this was very much a microscopy based examination, and they had a particular GPCR, CXCR4 as a chemokine, and they were using microscopy-based techniques to look at different expression levels and look at what happened with the receptor. And they found that with CXCR4, it really depends on the expression level, what type of oligomer it is. But I think this really depends on the types of membrane protein, what's going on. I don't think it's going to be a general thing.
No, fair enough. And then the other question around generalization I had, I mean, do you have to, how generalizable is the SMALP approach?
The SMALP approach really, again, different membrane proteins, it works in different ways. And there are, SMALP is just one of many different types of synthetic polymer. There's other polymers called DIBMALP. So it depends on styrene malic acid lipid particles as SMALP, and there are lots of others. I don't have the chemistry knowledge to go much more into it, but I do know that they allow you to have different size punches, and the different size might actually affect how constrained your membrane protein is. But the hope is that you might actually pull out your membrane protein and also pull out the partners that it's got that it normally associates with. I think it really much depends on the receptor, and you also have to build up a protocol. But the science, I think, is building, and there's some great websites out there that people have already done this technique for the particular membrane protein to make it easier.
Brilliant. Ben asks, I think with respect to the stem cells, she was wondering what the source of those stem cells were. Were they embryonic or induced, or what do you know where they came from?
These were Hues cells, so they're human embryonic stem cells, yeah. So they've been part of a program grant with the University of Nottingham. And I think they're very much a cardiovascular lab who are interested in trying to develop models to understand what's actually going on, so we can replace using just biopsies, which are, as you can understand, are quite hard to get hold of.
Brilliant. Awesome. Are there any other questions in the room? In which case, let us thank Joëlle again for a fantastic, exciting seminar. And we look forward to seeing you all in a couple of months' time for the next one. Watch your emails and the website and your socials for all the details. And we'll see you all soon. Thanks, Joëlle.
Thank you.